Hi everyone, I'm Sarath. I'm a doctoral student at Kerala Forest Research Institute, Kerala, India. So I would like to discuss on today on palms in Southern Asia, underrepresented genome hinders the conservation efforts. So when it comes to palms, there are pantropical monocotyledon lineage, which is diversified with approximately 2,600 species, which is functioning as a critical structural and functional component of tropical ecosystem, and they have been distributed in a wide variety of habitat from arid habitat to marshy lands. So this palm family is highly threatened by habitat loss, harvesting pressure, and invasive species spread. But IUCN only assessed and categorized 38% of the palm species, and of these, 17% are critically endangered, 18% are endangered, and 15% are vulnerable. When it comes to palms in Southern Asia, approximately 43 genera were diversified with 352 species. They were distributed in a wide variety of habitat, and an immense level of endemism and evolutionary distinct species were found in the biodiversity hotspots of Southern Asia, like Western Ghats, Himalaya, Indo-Burma, Sundaland, Wallacea, and Japan. And this plant family have high significance in the livelihood of native communities of this tropical region. So the plant family itself is identified as an economically important plant group. In Southern Asia, rattans, or also known as climbing palms, were highly traded globally. The global trade of the rattan were estimated to be 0.38 billion USD per annum in 2007. Other than this canes, many species were harvested for seeds, shoots, and leaves. So other than this harvestation pressure, many species were losing their habitat. Around 68 species were losing their habitat according to IUCN. And most of the species were losing their mutualistic species involved in the dispersal and persistence of palms. So conservation efforts are there, but most of the conservation efforts were mainly focused on the species level biodiversity, restoring species level biodiversity rather than conserving, but not focused on conserving intraspecific genetic diversity. So in a climate change scenario, the understanding on genetic diversity, adaptive potential, and the distribution of adaptive alleles in the landscape is a prerequisite for developing proper conservation plans. So one of the intents in this developing such conservation actions in palms is the lack of genomic information. So as an initiative, we have sequenced and developed a 592 billion base pair draft genome of Korthalsis laciniosa, which is actually a climbing palm in Andaman and Nicobar Islands in the Bay of Bengal. Other than this, Korthalsis species, Metroxylon sagu and Calamus simplicifolius is only two species which is revealed their genome. So along with the draft genome, we have also developed the Chloroplast genome. So this genome were used to mine SSRs. From the genome, we have identified approximately 2.5 lakh of SSRs, and we have identified 3,303 EST-SSRs from the CDS library. And from Chloroplast genome, we have identified 51 Chloroplast SSRs. So these SSR or these markers, genomic markers, will be helpful to identify and trace out the neutral evolutionary process and adaptive variation in the landscape in the species, which is actually evident from the photograph here shown. Each photograph shows different individuals in different populations in different islands. So this this variation and adaptive variation could be traced. So from the genomic SSR, we have developed and standardized 36 neutral genomic SSRs, which could be used to trace neutral genetic variation in the landscape, which is actually influenced by the landscape variables, demography, and the influence of anthropogenetic activities. So, and also some of the markers shown here were cross-transferred to Korthalsia rogersi and another vanishing threatened species of these islands. And from the CDS data, we have identified 3,303 microsatellites, and from which we have identified 24 SSR loci associated with the genes involved in stress and environment, which is actually shown here. And these markers could be helpful to trace the local adaptive variation, how each populations were adapted to different microclimate, and which all alleles or gene loci are potential for conserving and reinforcing the population to face the climate change. So from the chloroplast genome, we have identified 50 microsatellite loci, and we have developed 10 SSR loci, which is highly polymorphic. And using these markers, we will trace out the effect of defaunation. Actually, the islands were undergone a high deforestation, which affected the avian faunal assemblage. So these markers will be traced the effect of defaunation on the genetic characteristics of the palms of characteristics of these populations. So using these three markers, we will find out how the neutral evolutionary process, adaptive variation were structured in these islands, and we will find out how the adaptive variation can be used to reinforce and develop resilient conservation strategy in order to protect the non-timber forest product. Yeah, thank you.